Hello there, and welcome to the Sleepy Science Society. I'm so glad you found your way here tonight. Maybe you've had a long day filled with tasks and obligations, or perhaps you're simply the kind of person who prefers falling asleep to the gentle rhythm of scientific wonder rather than counting sheep or tossing restlessly. Either way, you've arrived at exactly the right place. Tonight's journey takes us into one of nature's most enchanting phenomena, a spectacle that transforms the darkest corners of our world into living galleries of light. You have nothing to do right now but rest. Let your shoulders drop, your breathing slow, and feel your body settle into comfort, as if gravity itself has become softer, more forgiving. Because tonight, we're exploring bioluminescence, the extraordinary ability of living organisms to produce their own light through chemical reactions. From the deepest ocean trenches where sunlight has never reached, to summer meadows dotted with fireflies, to hidden caves glowing with threads of blue-green radiance, we'll discover how life itself has learned to shine. This is a story written in light and chemistry, in evolution and survival, in beauty that seems almost too magical to be real. We'll meet creatures that hunt with living lanterns, others that disappear by glowing, and some that speak to one another in flashes and pulses across vast distances. We'll learn about chemical reactions so efficient they produce almost no heat, about symbiotic partnerships between animals and glowing bacteria, and about the surprising prevalence of this phenomenon in the largest habitat on Earth, the deep ocean. Before we begin our gentle descent into these illuminated depths, if you find these calm explorations help you unwind or provide a peaceful way to end your day, I'd truly appreciate it if you'd consider liking this video and subscribing to the channel. It helps others discover this quiet corner of science and wonder, one sleepy soul at a time. And now, let's dim the lights and begin our journey into the glowing darkness. The chemistry of living light. At the heart of every bioluminescent display, from the faintest glimmer to the brightest flash, lies a surprisingly simple chemical reaction. Yet, within this simplicity, exists an elegance that has captivated scientists for centuries. To understand bioluminescence, we must first understand the molecules that make it possible, and the dance they perform to create light in living tissue. The process begins with a molecule called luciferin. This is not a single compound, but rather a generic name given to any light-emitting molecule used in bioluminescence. The name itself comes from Lucifer, meaning light-bearer in Latin, though the molecules themselves are far less ominous than their namesake suggests. Luciferins are organic compounds that, when oxidized, release energy in the form of visible light. What makes them special is their molecular structure, which allows them to enter an excited state when they react with oxygen, then release that energy as photons as they return to their ground state. But luciferin doesn't perform this transformation alone. It requires a partner, an enzyme called luciferase. Like all enzymes, luciferase acts as a catalyst, dramatically speeding up the reaction between luciferin and oxygen. Without luciferase, the reaction would occur too slowly to be useful, if it occurred at all. The enzyme cradles the luciferin molecule, positioning it perfectly to interact with oxygen, and in doing so, orchestrates the creation of light. When these two molecules come together in the presence of oxygen, something remarkable happens. The luciferase enzyme helps bond the luciferin to oxygen molecules, creating an unstable intermediate compound in an excited energy state. This excited molecule is desperate to return to stability, and when it does, it releases the excess energy as a photon of light. The molecule that remains is called oxyluciferin, now chemically inactive until the organism replaces or recycles it. The luciferase enzyme, however, emerges unchanged from the reaction, ready to facilitate the process again and again, as long as luciferin and oxygen are available. This reaction produces what scientists call cold light, a term that perfectly captures one of bioluminescence's most remarkable features. Unlike the light from a candle flame or an incandescent bulb, where much of the energy is lost as heat, bioluminescent reactions are extraordinarily efficient. Less than 20% of the energy released becomes heat, with the vast majority converted directly into light. This efficiency is something human technology still struggles to match, even with our most advanced LED systems. But here's where the story becomes even more fascinating. There is no single luciferin molecule shared across all bioluminescent organisms. Instead, nature has invented this solution independently, over and over again, using different chemical structures to achieve the same glowing result. Scientists have identified at least 8 distinct types of luciferin molecules, each with its own unique chemical structure. Each evolved separately in different branches of the tree of life. Firefly luciferin, for instance, is completely different from the coelenterazine used by many marine organisms. The luciferin found in certain bacteria bears no chemical resemblance to that used by Dinoflagellates. And the light-producing molecules in bioluminescent fungi are different still. This diversity tells us something profound about bioluminescence. It is not an ancient trait inherited from a common ancestor. Instead, it has evolved independently at least 40 different times throughout the history of life on Earth, and possibly many more times than that. Each time bioluminescence evolved, organisms found their own chemical solution to the challenge of making light. Some adapted existing enzymes, modifying molecules that originally served other purposes. Firefly luciferase, for example, evolved from a group of enzymes called acyl-CoA synthetases, which normally help process fatty acids. Through millions of years of natural selection, these enzymes were repurposed, refined, and transformed into molecular machines capable of producing light. The independent evolution of bioluminescence suggests something important. It must confer significant advantages to the organisms that possess it. Evolution doesn't repeatedly invent complex traits unless those traits improve survival and reproduction. The fact that bioluminescence has arisen so many times in so many different groups of organisms across terrestrial and marine environments tells us that the ability to make light is genuinely useful in numerous ecological contexts. Recent research has revealed just how widespread this independent evolution has been. In 2018, scientists discovered that ray-finned fishes, the most diverse group of vertebrates on Earth, evolved bioluminescence 27 separate times. 27 distinct evolutionary events, each producing the same remarkable result through different molecular pathways. When you consider that most organisms capable of bioluminescence live in environments humans rarely see in the deep ocean or within forest soils, the true extent of this phenomenon becomes staggering. The chemistry can become even more sophisticated. Some organisms don't produce a continuous glow, but instead create what scientists call photoproteins. These are prepackaged combinations of luciferin and oxygen, bound together with the luciferase enzyme in a stable complex, waiting like a coiled spring. The light remains dormant until triggered by the presence of specific ions, usually calcium. The moment those calcium ions appear, the photoprotein complex releases its energy all at once in a sudden, bright flash. This allows organisms to produce light instantly, without the delay of mixing separate chemicals, perfect for startle responses or rapid signaling. The colors produced by bioluminescent reactions depend on the specific luciferin-luciferase combination, the pH of the environment, and even the presence of other proteins that can modify the emitted light. Most marine bioluminescence appears blue or blue-green, with wavelengths between 440 and 520 nanometers. This isn't random. Blue light travels farther through seawater than any other color, making it the most practical choice for communication, hunting, or defense in the ocean. Red light, with its longer wavelength, is quickly absorbed by water molecules and essentially vanishes within a few dozen meters of the surface. On land, where air doesn't filter light the same way water does, we see more variety. Fireflies produce yellow, yellow-green, or orange light, depending on the species. The light from different firefly species can range from 550 to 580 nanometers, variations that likely help each species recognize its own kind during mating displays. One remarkable beetle larva, known as the railroad worm, produces light in 2 different colors. Its body glows green while its head glows red. This creature possesses 2 different luciferase enzymes that modify the same luciferin molecule in different ways, creating a 2-toned display that resembles the lights of an old-fashioned train. Understanding the chemistry of bioluminescence has implications far beyond pure scientific curiosity. These efficient, light-producing reactions have become invaluable tools in medical research, molecular biology, and biomedical imaging. Scientists use luciferase as a reporter gene to track the activity of other genes, to monitor the spread of cancer cells, to detect bacterial contamination, and to study fundamental processes inside living cells. The same chemical reactions that allow a firefly to find a mate on a summer evening now help researchers develop new treatments for diseases and understand the basic mechanisms of life. As we drift deeper into our exploration, keep in mind this fundamental truth: bioluminescence is not merely a curious footnote in the story of life. It represents one of evolution's most successful innovations, a solution to countless problems faced by organisms in dark environments. It is chemistry transformed into light, molecules orchestrating a performance that has played out in Earth's oceans, forests, and soils for hundreds of millions of years. The deep ocean's light show. If you were to descend into the ocean, sinking slowly beneath the waves, you would witness one of the most dramatic transformations in any environment on Earth. The bright, sun-dappled surface waters would gradually dim, the familiar blues and greens fading meter by meter, until you passed through a boundary into perpetual darkness. This is the realm where bioluminescence becomes not a curiosity, but a fundamental fact of existence. The ocean is divided into distinct zones based on the penetration of sunlight. The uppermost layer, from the surface down to about 200 meters, is called the epipelagic or sunlight zone. Here, enough light penetrates for photosynthesis, and the water glows with the scattered blues of refracted sunlight. Life here is abundant, visible, familiar. But below this sun-touched realm lies the mesopelagic zone, stretching from 200 meters down to 1,000 meters deep. Scientists call this the twilight zone, a fitting name for a region where light fades rapidly with every meter of descent. The amount of sunlight decreases exponentially with depth, and by the time you reach the bottom of this zone, only the faintest whisper of surface illumination remains, barely enough to detect even with sensitive instruments. Below 1,000 meters, we enter the bathypelagic zone, more commonly known as the midnight zone. Here, sunlight does not reach. The darkness is absolute, broken only by the occasional shimmer of bioluminescence. This zone extends down to about 4,000 tiebad meters. Deeper still lie the abyssal and hadal zones, regions of crushing pressure and profound darkness that represent the largest habitat on Earth by volume. You might think that in such darkness, far from the energy of the sun, life would be scarce. You might imagine a barren, empty realm, a marine desert devoid of activity. But you would be profoundly mistaken. The deep ocean teems with life, and that life glows. Scientists estimate that between 76 and 90% of all animals living in the mesopelagic zone possess some form of bioluminescent capability. Let that sink in for a moment. In the twilight zone of the ocean, bioluminescence is not the exception but the rule. The majority of creatures you might encounter can produce their own light. This prevalence makes bioluminescence perhaps the most common form of communication on planet Earth. When we think of communication, we typically imagine sound, the songs of birds, the calls of whales, human language spoken and written. But in the largest habitat on our planet, creatures speak to each other in light. They flash signals to potential mates, warn rivals away from territory, coordinate hunting strategies, and send distress signals, all through carefully controlled pulses and patterns of bioluminescence. The deep sea is vast beyond human comprehension. If we were to take all the land on Earth, all the forests and deserts and tundras and grasslands, and add to it all the shallow coastal waters and the sunlit surface ocean, we would still have less volume than the deep ocean occupies. The bathypelagic zone alone is larger than all other habitats combined. And in this enormous realm, where darkness reigns supreme, bioluminescence transforms the environment into something otherworldly. Picture yourself suspended in this midnight water. Above and below, nothing but black. No surface shimmer, no seafloor visible, just an infinite dark that presses in from all sides. Then, suddenly, a pulse of blue light appears in the distance. Then another. A string of lights blinks on nearby, outlining a shape you can't quite make out in the darkness. A shower of sparkles erupts as something small disturbs a cloud of plankton. Somewhere far off, a steady glow suggests a hunting predator, its lure dangling in the current. The darkness isn't empty after all. It's alive with light, a slow-motion fireworks display orchestrated by chemistry and survival. The predominance of blue and blue-green bioluminescence in the ocean is no accident. Light behaves differently in water than in air. As photons travel through seawater, they interact with water molecules and dissolved substances, and this interaction varies by wavelength. Red light, with its long wavelengths, is absorbed almost immediately. Even in the clearest ocean water, red light vanishes within a few meters of the surface. Orange fades quickly after that, then yellow. Green light penetrates somewhat deeper, but blue light with wavelengths between 450 and 495 nanometers can travel the farthest through seawater before being absorbed. For organisms trying to communicate or hunt in the deep ocean, using blue light simply makes practical sense. A blue bioluminescent signal can be seen from much farther away than a red or yellow one would be. Over millions of years, natural selection has favored bioluminescent systems that produce blue wavelengths, because those systems allow organisms to be seen by intended receivers while minimizing the energy wasted on light that would be absorbed within a few meters. This explains why the vast majority of deep-sea bioluminescence appears blue or blue-green. It's an evolutionary optimization, life adapting its chemistry to the physics of its environment. The few exceptions to this rule are themselves fascinating. A small number of deep-sea fish, including certain species of dragonfish, have evolved the ability to produce red light, and they are the only organisms in their environment that can see it. We'll explore this remarkable adaptation in detail later, but for now, simply note that breaking the blue light rule requires not just producing a different color, but also evolving specialized eyes capable of detecting that color in an environment where it shouldn't exist. The deep ocean's light show is powered by thousands of different species, each with its own bioluminescent strategy. There are jellyfish that pulse with ghostly blue light, squid covered in patterns of photophores like constellations, tiny crustaceans called copepods that produce startling flashes when disturbed, bacteria that glow steadily, whether free-floating or housed within the light organs of larger animals. Countless species of fish, from the tiniest lanternfish to massive gulper eels, each with their own arrangement of light-producing organs. These photophores, the specialized light-producing structures found in marine animals, come in remarkable variety. Some are simple clusters of cells that produce a diffuse glow. Others are sophisticated organs complete with reflectors to amplify and direct the light, lenses to focus it, color filters to modify its wavelength, and even shutters of dark tissue that can be drawn across the photophore to turn the light on and off. The complexity of some photophores rivals that of the animal's own eyes. Tiny organs evolved specifically to control the production and emission of light with extraordinary precision. Many deep-sea fish have photophores arranged along their ventral surface, their bellies and undersides. Others have them scattered across their bodies in species-specific patterns. Some have a single large photophore under each eye, like headlights. The anglerfish, perhaps the most famous bioluminescent creature, has a single modified dorsal spine that extends forward like a fishing rod, with a glowing lure at its tip. Each species has evolved its particular arrangement in response to its particular needs, whether hunting, hiding, or finding mates in the dark. For organisms living in the twilight zone, where faint traces of sunlight still filter down from above, photophores serve an additional crucial function: camouflage. It might seem counterintuitive that producing light could help you hide, but in the gradual transition between light and dark, bioluminescence can make you invisible. Many fish in the mesopelagic zone use their ventral photophores to match the dim light coming from above, erasing their silhouette when viewed from below. This technique, called counterillumination, is so effective that it renders the fish nearly invisible to predators hunting from deeper waters. We'll explore this remarkable strategy in detail in our next section. The prevalence of bioluminescence in the deep ocean has profound implications for how we understand marine ecology. For decades, scientists studied the ocean primarily by pulling up samples in nets, examining what they caught, and making inferences about deep-sea life based on these collected specimens. But nets don't capture behavior. They don't reveal how organisms interact, how they hunt, or how they avoid being hunted. The development of deep-sea submersibles and remotely operated vehicles equipped with sensitive cameras has revolutionized our understanding, allowing researchers to observe bioluminescence in its natural context for the first time. What they've discovered is a world far more dynamic and interconnected than previously imagined. Bioluminescence isn't just a neat trick a few specialized species employ. It's a fundamental part of the deep-sea ecosystem, woven into the fabric of nearly every interaction. Predators hunt with light. Prey defend themselves with light. Potential mates find each other through light. The entire community is bound together by chains and webs of bioluminescent signaling, a vast communication network operating in the dark. As you rest and let your mind drift through these dark waters, imagine the scale of this phenomenon. Right now, as you listen, countless trillions of organisms in the deep ocean are producing light. Some are flashing to attract mates. Others are glowing steadily as they hunt. Still others are releasing clouds of luminous material to confuse predators. This has been happening every moment of every day for millions of years, an eternal light show in the largest habitat on Earth, mostly unseen by human eyes, playing out in the profound darkness where life has learned to make its own stars. Masters of disguise, camouflage through light. One of the most elegant and counterintuitive uses of bioluminescence in the ocean involves making yourself invisible by glowing. This might seem like a paradox. After all, isn't the whole point of camouflage to avoid being noticed? How can producing light possibly help you hide? But in the specific environment of the mesopelagic zone, where faint sunlight filters down from above but darkness surrounds you from all sides, bioluminescence becomes the key to the most effective camouflage in the animal kingdom. The problem faced by any animal swimming in the twilight zone is this. If you position yourself between a predator below and the faint light from the surface above, you become a dark silhouette, a shadow against the less dark water above you. Your body blocks this downward traveling light, creating a contrast that makes you highly visible to anything looking upward. For predators hunting from the depths, this silhouette stands out like a signal flare, an easy meal outlined against the faint glow of the world above. This creates an evolutionary pressure that has driven many mesopelagic fish to develop one of nature's most sophisticated camouflage techniques: counter-illumination. These fish have photophores arranged along their ventral surfaces, their bellies and undersides, precisely positioned to produce light that matches the intensity and color of the dim sunlight filtering down from above. By carefully controlling the brightness of their ventral photophores, these fish can eliminate their silhouette entirely, blending seamlessly with the background light when viewed from below. The precision required for effective counter-illumination is extraordinary. The light from above changes constantly as the sun moves across the sky, as clouds pass overhead, as the fish itself changes depth. Too much light from the photophores, and the fish would glow brighter than its surroundings, making it visible. Too little, and the dark silhouette would return. The fish must continuously adjust the intensity of its bioluminescence to match the ambient light conditions, effectively becoming transparent through careful application of its own illumination. Lanternfish, one of the most abundant vertebrates on Earth, have mastered this technique. These small fish, typically only a few inches long, possess rows of photophores along their ventral surface, each capable of being individually controlled. As a lanternfish swims through the twilight zone, it constantly monitors the light from above using specialized photoreceptor cells, then adjusts its photophore output to match. The result is remarkable. When viewed from below, a counter-illuminating lanternfish simply disappears, its outline erased by its own carefully modulated glow. But counter-illumination faces challenges. The dim light from the surface isn't uniform in color. It contains a spectrum of wavelengths, though blue predominates at depth. For truly effective camouflage, a fish needs to match not just the brightness but also the color spectrum of downwelling light. Some species have evolved photophores that can produce light in multiple wavelengths, or that incorporate color filters to modify their bioluminescence to better match ambient conditions. This adds another layer of complexity to an already sophisticated system. There's also the matter of viewing angle. Counter-illumination works beautifully when viewed from directly below, but what about predators approaching from the side or from above? Some fish have addressed this by arranging photophores, not just on their ventral surface but along their sides as well, creating a three-dimensional light envelope that helps conceal their form from multiple angles. Others have evolved body shapes that minimize their profile from side views, compressing themselves into thin, deep-bodied forms that present the smallest possible target to lateral viewers. The squid Abralia verani has taken counter-illumination to another level. This species lives in the mesopelagic zone and possesses photophores not just on its mantle, but also within its eyes. Yes, This squid has light organs in its eyes. The photophores are positioned in such a way that they fill the squid's pupils with light, eliminating the dark spots that would otherwise be visible from below. Even the eyes, which in many animals are a telltale sign giving away their position, become part of the camouflage system. As effective as counter-illumination is, it has a significant limitation. It only works in the twilight zone, where there's still some ambient light to match. Once an organism descends into the midnight zone, where no sunlight penetrates at all, counter-illumination becomes useless because there's no background light to blend with. Below about 1,000 meters, different strategies are required. In the midnight zone, many organisms have abandoned camouflage altogether and instead evolved to be utterly, completely black. Not just dark, but black in a way that absorbs nearly all light that hits them. Some deep-sea fish have skin so black that it absorbs 99.5% of visible light, making them essentially invisible even when bioluminescent light shines directly on them. This ultra-black coloration works by incorporating structures that trap light rays in a maze of melanin-filled compartments, preventing reflection. It's the opposite strategy from counter-illumination, achieving invisibility through perfect absorption rather than perfect matching of ambient light. But for organisms living at the boundary between light and dark, in those crucial 100 to 1,000 meter depths, counter-illumination remains one of evolution's most elegant solutions to the problem of predation. It demonstrates that bioluminescence is not merely about producing light for communication or hunting. It can also be about controlling how you appear to others, about manipulating the visual environment to your advantage, about using light to create darkness where your body would otherwise cast a shadow. The fish that have mastered counter-illumination carry with them a complete light management system. They have sensors to detect ambient light, neural circuits to process that information and calculate the required photophore output, and precise muscular control over their light organs to produce exactly the right intensity. They perform this adjustment constantly, in real time, as they swim through a world where the lighting conditions can change significantly over just a few meters of vertical movement. Consider the engineering challenge from a human perspective. We've developed camouflage systems for military vehicles and vessels that attempt to match their surroundings. But these are crude approximations compared to what a simple lanternfish achieves. Our best adaptive camouflage requires cameras, processors, and active display systems, consuming significant power and still falling short of perfect invisibility. A lanternfish, with a brain smaller than a grain of rice, continuously solves this problem using nothing but biological tissues and chemical reactions, maintaining perfect camouflage while simultaneously swimming, navigating, finding food, and avoiding predators. As we drift through these concepts, imagine yourself as one of these counter-illuminating fish. You hang suspended in the twilight zone, the faint glow of the surface far above, the absolute darkness of the abyss far below. You feel the photoreceptors in your skin detecting the subtle changes in ambient light as a cloud passes over the surface hundreds of meters above you. Automatically, without conscious thought, your ventral photophores dim slightly to match. You are both there and not there. A ghost in the water, visible only to those who know how to look beyond the apparent emptiness. You have become living proof that sometimes the best way to hide is to glow just enough to disappear. Hunters in the dark, in the perpetual night of the deep ocean, finding food presents an extraordinary challenge. Prey is sparse, scattered across vast distances in a three-dimensional space. Swimming constantly to search for food wastes precious energy in an environment where meals are few and far between. Many deep-sea predators have solved this problem by becoming ambush hunters, waiting motionless in the dark for prey to come to them. But how do you lure prey in an environment of total darkness? The answer for many species is bioluminescence used as a hunting tool, a living lure that draws curious prey within striking distance. The most famous of these bioluminescent hunters is undoubtedly the deep-sea anglerfish. These fish have evolved one of the most distinctive body plans in the ocean. A massive head dominated by a huge, fang-filled mouth, a compressed body, and most remarkably, a modified dorsal spine that extends forward like a fishing rod. At the end of this spine, called the elysium, dangles a fleshy lure known as the esca, and within this esca lives a colony of bioluminescent bacteria. The relationship between an anglerfish and its bacterial partners is a marvel of evolutionary cooperation. The bacteria belong to the genus Photobacterium, and they produce light continuously as a byproduct of their metabolism. The anglerfish provides these bacteria with a safe home and a steady supply of nutrients through blood vessels that perfuse the esca. In return, the bacteria provide the anglerfish with its most important hunting tool, a glowing lure that attracts curious prey in the darkness. The esca is not just a simple blob of glowing tissue. It's a sophisticated light organ with structures that amplify and direct the bacterial light. Some species have a reflective layer behind the bacteria to increase the forward projection of light. Others have a dark covering that can be drawn over the esca like a shutter, allowing the anglerfish to turn its lure on and off. This on-off capability is crucial for hunting. A constantly glowing lure might attract not just prey, but also predators or competitors. By controlling when the lure is visible, the anglerfish maintains tactical advantage. The hunting strategy is simple but effective. The anglerfish positions itself motionless in the water column, often oriented slightly upward. Its enormous mouth remains closed, its body nearly invisible in the perfect darkness. Then it activates its lure, revealing a small, glowing point of light that dangles and sways with the motion of the water. To a small fish or crustacean swimming through the midnight zone, this light resembles food, perhaps a piece of bioluminescent plankton or a small glowing organism. Curious, hungry, or simply attracted by the only visible object in an otherwise featureless dark, the prey approaches to investigate. The prey never sees the anglerfish until it's too late. The massive jaws are dark and perfectly camouflaged, invisible against the black water. The glowing esca, positioned at the end of its rod-like elysium, draws attention away from the body. By the time the prey realizes the light is attached to something much larger, the anglerfish strikes. Its mouth opens with shocking speed, creating a powerful suction that draws the prey inside along with a rush of water. The entire attack can be over in a fraction of a second, and the anglerfish returns to its motionless waiting, its lure ready to draw in the next victim. Different species of anglerfish have evolved variations on this basic theme. Some have elaborate, branching elysia with multiple lures. Others have bioluminescent barbels hanging from their chins in addition to the main lure. The female anglerfish, which are the ones bearing the lures, can grow to sizes ranging from a few inches to more than 3 feet long, depending on the species. The males, in one of nature's most extreme examples of sexual dimorphism, are tiny parasitic partners that bite onto the female and fuse their circulatory systems, essentially becoming a permanent sperm-producing appendage. But that's a story for another time. The anglerfish isn't the only deep-sea predator that has weaponized bioluminescence for hunting. The dragonfish, a family of elongated predators with fang-like teeth, have evolved a different and perhaps even more remarkable strategy. Many dragonfish possess a specialized photophore located just below their eye, essentially a biological spotlight. But unlike most bioluminescent organs in the ocean, which produce blue or blue-green light, the dragonfish's suborbital photophore produces red light. This red light production is extraordinary because, as we've discussed, red light is rapidly absorbed by seawater, and most deep-sea organisms cannot see it. The photoreceptors in fish eyes are typically tuned to blue wavelengths, the dominant color of bioluminescence and the color that travels farthest through water. Red wavelengths might as well not exist as far as most deep-sea creatures are concerned. But dragonfish have evolved specialized visual pigments that allow them to detect red light. They are essentially using a form of light that only they can see, a private wavelength invisible to everything around them. The tactical advantage this provides is immense. A dragonfish can illuminate its surroundings with its red photophore, lighting up potential prey without alerting them to its presence. The prey, unable to see red light, remains oblivious as the dragonfish examines it in detail, judges its size and health, and positions itself for an attack. It's the equivalent of a soldier using infrared night vision goggles in combat, seeing clearly in a spectrum invisible to the enemy. What makes this even more remarkable is that dragonfish accomplish red light production using chlorophyll derivatives, molecules typically associated with photosynthesis in plants. These chlorophyll-based compounds, incorporated into the eye itself, allow dragonfish to detect wavelengths that no other deep-sea predators can sense. The evolutionary pathway that led to this innovation remains partially mysterious, but its effectiveness is undeniable. The loosejaw dragonfish, a particularly specialized group, have taken this strategy even further. These fish have evolved lower jaws that lack a connecting membrane, allowing their mouths to open incredibly wide relative to their body size. Combined with their red light vision, this means loosejaws can detect and consume prey much larger than themselves, gulping down meals that would be impossible for a more conventionally built fish of the same size. Not all bioluminescent hunting strategies involve lures or spotlights. Some predators use bioluminescence more subtly, as a form of active camouflage that allows them to approach prey undetected. The cookie-cutter shark, a small but fearsome predator, has photophores covering most of its ventral surface. These photophores help it achieve counter-illumination when viewed from below, making it nearly invisible as it swims beneath its prey. But the cookie-cutter shark has a dark patch on its throat that does not bioluminesce. When viewed from below, this dark patch stands out against the glowing belly, resembling a small fish. Larger predators, attracted by what appears to be an easy meal, approach to investigate. When they get close, the cookie-cutter shark strikes, not to kill, but to take a bite. Its specialized teeth allow it to excavate a circular plug of flesh from the side of the victim, leaving a distinctive crater-like wound. The shark gets a nutritious meal, and the victim, though injured, usually survives. It's a parasitic strategy that uses bioluminescent camouflage in reverse. Being visible in just the right way to attract curious predators close enough to become victims themselves. Viperfish, another group of deep-sea predators, have enormous fangs and photophores lining their bodies. Their hunting strategy appears to involve using their body photophores to provide just enough light to see their prey in the final moments before an attack. The light also serves to startle or disorient prey, making escape more difficult. With teeth so large they cannot fully close their mouths, viperfish are built for impaling prey rather than biting, and their bioluminescence helps them position their fearsome dental arrays with deadly accuracy. As we explore these predators and their glowing hunting tools, a pattern emerges. Bioluminescence in the deep sea is not a simple phenomenon with a single purpose. It's a toolkit, a collection of strategies that different species have adapted to their particular ecological niches. For the anglerfish, light is a lure. For the dragonfish, light is a spotlight in a private wavelength. For the cookie-cutter shark, light is camouflage with a calculated flaw that turns defense into offense. Each predator has found its own way to weaponize chemistry, transforming the production of photons into a mechanism for capturing the energy stored in other living bodies. These hunting strategies also highlight the evolutionary arms race between predators and prey in the deep ocean. As predators evolved more sophisticated bioluminescent hunting tools, prey species evolved counter-strategies. Some became sensitive to the wavelengths commonly used in bioluminescent lures, learning to avoid rather than approach certain patterns of light. Others evolved to move in unpredictable patterns, making them harder to target even once illuminated. Still others developed their own defensive bioluminescence, which we'll explore in the next section. The deep ocean is an environment of extreme selective pressure. Food is scarce, energy must be conserved, and any innovation that improves hunting success or predator avoidance can mean the difference between evolutionary success and extinction. Bioluminescent hunting represents millions of years of refinement, of trial and error written in genes and expressed in chemistry. These fish are not conscious engineers, but the systems they've evolved demonstrate a level of sophistication that human technology struggles to replicate. They are hunters adapted to hunt in perfect darkness, using living light as their primary weapon. Defense mechanisms, light as protection. If bioluminescence can be used as a weapon, it can certainly be used as a shield. Indeed, some of the most spectacular displays of bioluminescence in the ocean occur not during hunting, but during defensive responses to predation attempts. When the stakes are survival, organisms have evolved startlingly creative ways to use light to confuse, scare, or distract attackers, buying precious seconds for escape. The vampire squid, despite its ominous name, is not actually a fearsome predator, but rather a gentle scavenger that feeds on marine snow and detritus drifting down from the upper ocean. Its name comes from its dark coloration and the webbing that connects its 8 arms, which can be flipped inside out to create an appearance reminiscent of a vampire's cape. But what truly sets this remarkable cephalopod apart is its sophisticated defensive use of bioluminescence. When threatened by a predator, the vampire squid has several defensive options. It can curl its arms up and over its body, exposing rows of spiky protrusions called cirri, while simultaneously tucking its most vulnerable parts, its head and internal organs, safely away inside the protective embrace of its arms. In this posture, called the pumpkin or pineapple position, the vampire squid presents a much larger, more intimidating profile, and those cirri look distinctly unappetizing. But the vampire squid's most spectacular defense involves bioluminescence. When highly agitated, it ejects clouds of sticky bioluminescent mucus from the tips of its arms. This isn't a simple puff of glowing material. The mucus contains thousands of tiny orbs of blue light, each one about the size of a pinhead, that persist for several minutes, creating a shimmering, confusing cloud in the water. The effect is disorienting. A predator pursuing the squid suddenly finds itself surrounded by hundreds of glowing points, making it difficult to track which direction the squid went or even to maintain focus on any particular target. While the predator is distracted by this spectacular light show, the vampire squid escapes into the darkness. The glowing mucus also sticks to the predator, making it more visible to its own predators. A fish covered in bioluminescent slime stands out like a beacon in the deep ocean, attracting unwanted attention from larger predators higher up the food chain. This burglar alarm effect turns the tables on the attacker, transforming it from hunter to hunted. The vampire squid's photophores, the light-producing organs scattered across its body, can also be individually controlled. It can pulse them in patterns, creating waves of light that ripple across its body surface. It can turn them on and off like Christmas lights, as one researcher memorably described. This precise control allows the vampire squid to create complex visual displays that may serve multiple functions, from startling predators to communicating with potential mates. Though much about this species remains mysterious due to the difficulty of observing it in its natural deep-sea habitat. Many species of squid and shrimp employ similar defensive bioluminescence strategies. The deep-sea shrimp Acanthophera purpurea, when threatened, can spew bioluminescent chemicals from glands near its mouth, creating a glowing vomit-like substance that confuses predators. This is sometimes called spew bioluminescence, and it functions like a bioluminescent smokescreen. The shrimp literally throws light into the water, creating a distraction while it jets backward into the safety of darkness. Some species of brittle stars, relatives of sea stars that live in deep and shallow waters, produce bright flashes of light when disturbed. The exact function of this bioluminescence isn't entirely clear, but researchers believe it may serve as a warning signal, essentially announcing to predators that the brittle star is unpalatable or toxic. This aposematic use of bioluminescence, using light to advertise danger, is less common in the ocean than on land, but still exists among certain species. The burglar alarm strategy deserves special attention because it's so counterintuitive. When attacked, some organisms don't try to hide or run. Instead, they light up as brightly as possible, creating a bioluminescent beacon that attracts the attention of larger predators. The Atolla jellyfish demonstrates this beautifully. When attacked, it produces a spectacular rotating display of bioluminescent pulses that race around the rim of its bell. This alarm wheel display is thought to attract larger predators that might prey on whatever is attacking the jellyfish. Essentially, the jellyfish is calling for backup, using light to summon its attacker's predators. This strategy works because the deep ocean has complex food webs with multiple trophic levels. Small fish eat small jellyfish. Medium fish eat small fish. Large fish eat medium fish. By creating a highly visible display when attacked, the jellyfish increases the probability that a larger predator will notice and investigate, potentially attacking the small fish that was attacking the jellyfish. The jellyfish might still get eaten, but it has a chance, and in evolutionary terms, any strategy that occasionally works is better than none. Some species have evolved the ability to detach bioluminescent body parts when attacked. Certain squid can shed the tips of their arms, which continue to glow after detachment. The predator, drawn to the brightest and most active thing in its vicinity, attacks the arm tip while the squid escapes in the opposite direction. The arm tip eventually regenerates, a small price to pay for survival. This sacrificial bioluminescence strategy is particularly effective because the detached arm tip often continues to move and pulse with light, creating a convincing decoy. Scale worms, segmented marine worms found at various depths, can shed their scales when attacked. These scales continue to produce bioluminescent flashes after being released, distracting predators. Some species of brittle stars can detach entire arms that writhe and glow, drawing predatory attention away from the main body. These sacrificial strategies represent a fascinating cost-benefit calculation. Growing new body parts requires energy and time, but being eaten requires everything. The evolutionary math favors species that can sacrifice a part to save the whole. Firefly larvae, the terrestrial relatives of these marine bioluminescent organisms, also use light defensively. Many firefly larvae are unpalatable due to toxic compounds in their bodies, and their steady glow serves as a warning to potential predators. Birds and other predators that bite a glowing firefly larva quickly learn to associate that light with an unpleasant taste and avoid similar larvae in the future. This aposematic use of bioluminescence is common among terrestrial bioluminescent insects, where the light functions as an honest signal, warning predators away from a genuinely unpleasant meal. The diversity of defensive bioluminescence strategies reflects the intense predation pressure in environments where these organisms live. In the deep ocean, where food is scarce, every animal is constantly balancing the need to find food with the need to avoid becoming food. Bioluminescence has been shaped by this pressure into a sophisticated defensive toolkit. Some defenses are passive, like the warning glow of toxic species. Others are active, requiring the organism to deploy bioluminescence at exactly the right moment to maximize its chance of survival. What makes these defensive strategies particularly remarkable is how effectively they exploit the perceptual and neurological systems of predators. The vampire squid's glowing mucus cloud works because it overwhelms the predator's visual system with targets. The burglar alarm works because it exploits the predator's own fear of being eaten. Sacrificial glowing body parts work because predators are hardwired to track movement and light. These defenses aren't just chemical reactions. They're psychological warfare, evolution crafting solutions that manipulate the hunter's own instincts against it. As you rest and contemplate these strategies, consider how bioluminescence reveals the complexity of evolutionary solutions. The same chemical reaction, the oxidation of luciferin by luciferase, has been adapted to countless purposes. It lures prey, frightens predators, summons help, creates decoys, and advertises danger. Each organism has taken this basic toolkit and modified it, refined it, and deployed it in ways that increase survival in its specific environmental context. In the dark waters of the ocean, light itself has become a language of survival, spoken in flashes, pulses, clouds, and beacons, a chemical conversation about life and death written in photons. Communication and courtship light in the deep ocean isn't used solely for hunting and defense. Some of its most beautiful and complex applications involve communication between members of the same species, particularly during courtship. Finding a mate in the vast, dark expanse of the open ocean presents challenges that would be difficult to overstate. The ocean is enormous, and individuals of any given species are scattered across vast distances. In darkness, visual cues are limited. Sound travels well in water, but can attract unwanted attention from predators. Bioluminescence offers a solution. A way to signal across distance without the limitations of sound or the need for ambient light. Fireflies, the terrestrial masters of bioluminescent communication, have evolved perhaps the most sophisticated courtship light displays in nature. On warm summer evenings across much of the world, firefly males take flight, producing species-specific patterns of flashes as they patrol their territories. Each species has its own distinctive pattern. The duration of each flash, the interval between flashes, the number of flashes in a sequence, even the color of the light, all vary from one firefly species to another. Female fireflies watch from vegetation, waiting for a male of the correct species to fly past. When a female sees the right pattern, she responds with her own flash after a precisely timed delay. This delay is crucial. For Photinus pyralis, a common North American firefly, the female waits approximately 2 seconds after the male's flash before responding. This exact interval tells the male that he's found a receptive female of his own species. The male, seeing the response, changes course and flies toward her. The exchange continues. A light-based dialogue, until the male locates the female and mating occurs. The precision of this system is remarkable. The timing must be exact. Flash too early or too late, and the male will assume you're the wrong species and continue searching. The pattern must match. Flash with the wrong rhythm or duration, and you're invisible to potential mates. This specificity helps maintain reproductive isolation between closely related species that might occupy the same habitat. Even though different firefly species may fly in the same meadow on the same night, each can identify and locate mates of their own kind through their unique bioluminescent signatures. But this system, like all communication systems, is vulnerable to exploitation. Some female fireflies in the genus Photurus have evolved the ability to mimic the flash responses of females from other species. When a male of the mimicked species approaches expecting to mate, the Photurus female instead attacks and eats him. These femme fatales use bioluminescent communication as a hunting strategy, exploiting the mating signals of other species to lure in prey. It's a dark twist on courtship communication, a reminder that any signal can potentially be faked if there's an evolutionary advantage to doing so. In the ocean, firefly squid provide an example of the coordinated bioluminescent displays during mating. These squid, which inhabit waters around Japan, gather in huge numbers during breeding season. The males produce synchronized, flashing patterns across their photophores, creating what looks like an underwater light show. These displays help females identify healthy, vigorous males and may also serve to intimidate rival males. During spawning aggregations, the waters can glow with the combined bioluminescence of millions of individual squid, each flashing in patterns that blend into a collective brilliance. Deep-sea anglerfishes face a unique challenge in mate finding. The female anglerfish, as we've discussed, can be quite large and uses her bioluminescent lure to hunt. The male, however, is tiny, sometimes only a few centimeters long, and lacks a lure. His entire life strategy is focused on finding a female. When he does, he bites onto her body, and in many species he never lets go. His tissues fuse with hers, their circulatory systems connect, and he essentially becomes a parasitic appendage, providing sperm when needed. But how does the tiny male find a female in the vast darkness of the deep ocean? The answer appears to involve species-specific bioluminescent patterns. Female anglerfish produce not just the lure on their heads, but also patterns of photophores on their bodies. These patterns vary by species and may help males identify appropriate females in the dark. The male's journey to find a female is a one-way trip. He doesn't feed as an adult, living off reserves accumulated during his larval stage. He must find a female before those reserves run out, or he will die. The bioluminescent beacon of the female is literally the male's only hope for reproductive success. Other deep-sea fish use bioluminescence for species recognition in less extreme ways. Many mid-water fish have species-specific arrangements of photophores, patterns as distinctive as fingerprints. These patterns allow individuals to recognize potential mates even in the dim light of the mesopelagic zone. Some species have photophores that produce different colors or that flash in particular rhythms, all serving to help individuals identify and choose mates of their own species. Certain ostracods, tiny crustaceans common in both shallow and deep waters, produce bioluminescent courtship displays that rival those of fireflies. Male Caribbean ostracods release bioluminescent secretions while swimming in distinctive patterns near the seafloor. The glowing trails they leave behind form species-specific shapes, rising helixes, or pulsing chains of light. Females, observing these displays from the seafloor, choose mates based on the quality and consistency of their light shows. The displays are ephemeral, lasting only seconds before the bioluminescent secretions fade. But in those brief moments, they convey information about the male's health, vigor, and species identity. Coral spawning, while not strictly bioluminescent, demonstrates another form of light-coordinated reproduction in the ocean. Many coral species spawn simultaneously, releasing eggs and sperm into the water in massive, synchronized events. While the eggs and sperm themselves aren't bioluminescent, the timing of these spawning events appears to be coordinated partly by light cues, including moonlight and the brief flashes of bioluminescent plankton that may signal optimal environmental conditions. The evolution of bioluminescent courtship signals represents a fascinating intersection of sexual selection and natural selection. Males must produce signals bright enough and distinctive enough to attract females, but not so bright that they attract predators. Females must be able to detect and evaluate male signals, distinguishing between species and between high-quality and low-quality potential mates, all while avoiding becoming prey themselves. The result is a delicate balance, an evolutionary optimization of signal intensity, pattern, and timing. In some species, bioluminescent displays serve double duty, functioning in both mate attraction and territory defense. Male dragonfish, for example, use their photophores both to attract females and to establish dominance over other males. The displays become a form of competitive signaling, with males attempting to outshine their rivals while simultaneously attracting female attention. This multi-purpose use of bioluminescence demonstrates its versatility as a communication tool. The study of bioluminescent communication has revealed fundamental principles about how signals evolve and how communication systems remain reliable. For a signal to be useful, it must be honest, conveying real information about the signaler. Firefly flash patterns honestly advertise species identity, because individuals that flash the wrong pattern fail to reproduce. The bioluminescent displays of healthy males are brighter and more consistent than those of sick or weak males, providing females with honest information about mate quality. Predators and parasites that exploit these signals, like the photourus fireflies that mimic other species, are exceptions that prove the rule. Communication systems are vulnerable to exploitation precisely because they usually provide reliable information. As you drift through these thoughts, imagine the scale of bioluminescent communication happening at this very moment. In oceans around the world, in forests and meadows on every continent where fireflies live, organisms are producing light to communicate. They're flashing species-specific patterns, attracting mates, establishing territories, and engaging in the eternal dance of reproduction. Each flash is a message, each pattern a conversation, each display a small victory in the ongoing challenge of finding connection in the vastness of the living world. Symbiotic relationships. Some of the most fascinating aspects of bioluminescence involve partnerships between species. Symbiotic relationships, where one organism provides the light while another provides the resources needed to make that light. These partnerships reveal the deeply interconnected nature of life, demonstrating how evolution can craft solutions that involve not just individual organisms, but entire communities of species working together. The Hawaiian bobtail squid and the bacterium Vibrio fischeri form perhaps the best-studied symbiotic bioluminescent partnership in marine biology. This tiny squid, only a few centimeters long, lives in shallow coastal waters around Hawaii and spends its nights hunting small crustaceans over sandy seafloors. During these nocturnal hunts, the squid needs to avoid being eaten by predators that might spot its silhouette against the moonlight filtering down from above. The squid solves this problem through bioluminescent counter-illumination, but remarkably, it doesn't produce the light itself. Instead, within hours of hatching, the young squid is colonized by Vibrio fischeri bacteria from the surrounding seawater. These bacteria take up residence in a specialized light organ in the squid's mantle, a chamber specifically evolved to house them. The organ provides the bacteria with nutrients, primarily amino acids, that the squid delivers through a rich network of blood vessels. The bacteria, in turn, glow continuously, producing blue-green light through the standard luciferin-luciferase reaction. But the squid doesn't need light during the day, when it buries itself in sand to rest. Continuous glow would waste energy and potentially attract predators. So each morning, as dawn approaches, the squid expels about 90% of its bacterial colony back into the ocean. During the day, the remaining 10% multiply, rebuilding the population. By nightfall, when the squid emerges to hunt, its light organ is fully repopulated and glowing at full intensity. The squid has even more sophisticated control over this bacterial light. The light organ includes structures that focus and direct the bacterial glow downward, matching it to the intensity of moonlight or starlight filtering through the water above. An ink sac sits above the light organ, functioning like an adjustable shutter that the squid can use to modulate light output. The system also includes reflective tissues that amplify the light and direct it in specific directions. The result is a perfectly calibrated counter-illumination system that renders the squid nearly invisible when viewed from below. This relationship between squid and bacteria benefits both partners. The bacteria receive a protected environment with steady nutrition, allowing them to reproduce far more successfully than they could in the open ocean. The squid receives a light source without having to synthesize its own luciferin and luciferase, which would require significant metabolic investment. The partnership is so successful that every Hawaiian bobtail squid carries this bacterial population, generation after generation. A living legacy passed not through genetics but through environmental acquisition and maintenance. Researchers have used the bobtail squid system to study fundamental questions about symbiosis. How do the bacteria and squid recognize each other? How does the squid's immune system distinguish between helpful Vibrio fischeri and harmful bacterial invaders? How do chemical signals coordinate the partnership? The answers have implications far beyond bioluminescence, shedding light on how microbiomes function, how organisms interact with beneficial bacteria, and how complex partnerships evolve. Many deep-sea fish have evolved similar partnerships with bioluminescent bacteria. The anglerfish, as we've discussed, houses bacteria in its esca, the dangling lure at the end of its elysium. The bacteria provide continuous light, and the fish provides nutrients and protection. But the partnership goes deeper than simple exchange. The anglerfish has evolved specialized tissues in the esca that reflect and focus the bacterial light, effectively turning a diffuse glow into a concentrated beam. It's a cooperation not just between species but between tissues and cells, each component optimized to enhance the effectiveness of the whole system. Different groups of deep-sea fish have independently evolved bacterial light organs multiple times. Flashlight fish, members of the family Anomalophidae, have large light organs beneath their eyes filled with bioluminescent bacteria. These fish can rotate the organ or cover it with dark tissue, effectively turning their flashlights on and off. They use these lights for communication, finding prey, and confusing predators by flashing the light, then quickly moving in a different direction, creating a false trail. Ponyfish, small coastal fish common in tropical waters, have light organs in their esophagi filled with symbiotic bacteria. The light shines down through the fish's body and out through its transparent ventral surface, providing counter-illumination. The fish can control light intensity by adjusting blood flow to the light organ, altering the oxygen supply available to the bacteria and thus modulating their glow. Pinecone fish have elaborate light organs in their lower jaws, filled with bacteria that glow red, orange, or yellow rather than the typical blue-green. These fish open and close their mouths to flash their lights, using the bioluminescence for communication and possibly to illuminate prey. The exact species of bacteria involved and the evolutionary history of this partnership remain areas of active research. Not all symbiotic relationships involving bioluminescence are as direct as bacteria living in specialized light organs. Some partnerships involve organisms that consume bioluminescent prey and then use the ingested luciferin for their own light production. Certain species of midshipman fish, for example, cannot synthesize their own luciferin. Instead, they obtain it by eating ostracods, tiny crustaceans that contain high concentrations of the molecule. The fish sequester this luciferin and use it in their own bioluminescent displays, essentially stealing the chemical raw materials from their food. This dietary acquisition of luciferin is more common than once thought. Many marine organisms appear to rely on consuming luciferin rather than synthesizing it themselves, creating food web connections where light-producing chemicals pass from prey to predator. These connections mean that bioluminescence isn't just an individual or species-level phenomenon, but a community-level one, with light-producing capabilities flowing through ecosystems like energy or nutrients. Symbiotic relationships involving bioluminescence also exist on land, though they're less common than in the ocean. Certain insects form partnerships with bioluminescent bacteria, though these are less well studied than marine symbioses. Some millipedes and beetles that live in caves or deep soil have bacterial partners that may contribute to their faint glow, though the exact nature of many of these relationships remains mysterious. The evolution of symbiotic bioluminescence raises fascinating questions about how such partnerships begin and how they're maintained over evolutionary time. For a symbiosis to evolve, both partners must benefit more from the partnership than they would independently. For bioluminescent symbioses, this means the host must provide enough resources to sustain the bacterial population while receiving enough light to justify that investment. The bacteria must be specialized enough to live in the host environment while remaining capable of producing light efficiently. Once established, these partnerships can drive further evolution in both partners. The host may evolve more sophisticated structures to house and control the bacteria. The bacteria may evolve to produce more light or to respond more precisely to host signals. Over time, the partnership becomes so integrated that neither partner can thrive without the other. The Hawaiian bobtail squid, for instance, develops normally even without its bacterial partners, but adults without functioning light organs have dramatically reduced survival rates because they cannot perform effective counter-illumination. These symbiotic partnerships also illustrate a fundamental principle of biology. Life doesn't exist in isolation. Every organism is part of a network of relationships, dependencies, and interactions that span multiple species and scales. The glow of a bobtail squid isn't just the squid's achievement, it's the product of a partnership refined over millions of years. The bacterial cells producing the light are, in a very real sense, part of the squid, integrated into its body and its life cycle, even though they belong to a completely different domain of life. As you rest here, considering these partnerships, think about how bioluminescence reveals the collaborative nature of evolution. Life doesn't solve every problem alone. Sometimes the best solutions involve bringing together different species, different capabilities, different evolutionary innovations, and combining them into something neither could achieve independently. The glowing lure of an anglerfish represents not just the fish's evolution, but also the bacteria's evolution and the millions of years of partnership that brought them together. Light, in these cases, is a shared language, a common currency, a bridge between species built from chemistry and sustained by mutual benefit. Terrestrial bioluminescence. While the ocean dominates any discussion of bioluminescence, the phenomenon is not exclusive to marine environments. On land, particularly in temperate and tropical forests, bioluminescence creates displays that are no less spectacular for being less common. The ecological functions, the organisms involved, and the evolutionary pressures differ significantly from marine environments. Yet the underlying chemistry remains remarkably similar. Another example of evolution discovering the same basic solution in dramatically different contexts. Fireflies and their close relatives, the glowworms, are the most visible and celebrated terrestrial bioluminescent organisms. These insects, all members of the beetle family Lampyridae, have evolved sophisticated light-producing organs and complex behavioral displays that rival anything found in the ocean. On summer evenings across Europe, North America, Asia, and parts of South America, fireflies emerge to create one of nature's most enchanting spectacles. The bioluminescent organs of fireflies are typically located in specialized segments of the abdomen called lanterns. These organs contain cells packed with mitochondria and peroxisomes, the cellular structures that produce energy and handle oxygen metabolism. Within these cells, firefly luciferin reacts with oxygen in the presence of firefly luciferase and ATP, the universal energy currency of cells. The reaction is remarkably efficient, with nearly 100% of the energy released appearing as light rather than heat. The color of firefly light varies by species, ranging from yellowish-green to orange-red. These color differences arise from variations in the luciferase enzyme and the pH of the environment where the reaction occurs. The same luciferin molecule can produce different wavelengths of light depending on how the luciferase holds it during the reaction and what other molecules are present. This variability has allowed different firefly species to evolve distinctive colors that help with species recognition. What makes fireflies particularly remarkable is their conscious control over their bioluminescence. Unlike the continuous glow produced by bacteria or the triggered responses of many marine organisms, fireflies can flash their lights on and off at will, with precise timing down to fractions of a second. They achieve this control through neural signals that regulate the supply of oxygen to the light-producing cells. When a firefly decides to flash, nerve impulses trigger the release of nitric oxide within the lantern. This chemical causes mitochondria to temporarily stop using oxygen for normal cellular respiration, freeing that oxygen to participate in the bioluminescent reaction instead. When the oxygen supply is cut off, the light stops almost instantaneously. This on-off control allows fireflies to produce complex temporal patterns, sequences of flashes with specific durations and intervals that convey information about species, sex, and individual quality. Male fireflies produce species-specific patterns as they fly, advertising their presence to females. Females respond from vegetation or the ground, typically with a single flash that comes after a precise delay. The male, detecting the response, flies toward the female, continuing to flash. If the female continues to respond at the correct interval, the male eventually locates her, and mating occurs. Different firefly species have evolved astonishingly different flash patterns. Some produce single, brief flashes at regular intervals. Others create double flashes or triplets. Some produce long, sustained glows rather than discrete flashes. A few species, particularly in Southeast Asia, have evolved synchronized flashing, where thousands of male fireflies in trees flash in perfect unison, creating a pulsing light display visible from great distances. The mechanism coordinating this synchronization involves individual fireflies adjusting their flash timing based on the flashes of their neighbors, gradually bringing the entire population into phase like biological oscillators finding resonance. Firefly larvae also produce light, though their bioluminescence serves a different function than the adult's courtship displays. Many firefly larvae are predators, hunting snails and other soft-bodied invertebrates in leaf litter and soil. Their steady glow appears to function as a warning signal, advertising the larvae's unpalatability to potential predators. Firefly larvae contain toxins called lucibufficins, bitter-tasting compounds that make them distasteful or even poisonous to birds and other predators. The bioluminescent warning is an honest signal. Predators that ignore the glow and eat the larva have an unpleasant experience and learn to avoid glowing larvae in the future. Glowworms, despite the name, are not worms but the larvae or larviform females of various beetle species. The most famous are found in caves in New Zealand and Australia, where they create spectacular displays. These glowworms hang sticky silk threads from cave ceilings to capture flying insects. Their bioluminescent glow attracts prey in the darkness of the cave, luring insects toward the threads where they become entangled. Once caught, the prey is consumed by the glowworm. These displays can be so extensive that entire cave chambers appear to have ceilings covered in blue-green stars, though each star is actually the glowing posterior end of a single larva. The railroad worm, which we've mentioned earlier, is the larva of a South American beetle. It possesses the remarkable ability to produce two colors of light: green photophores along its body segments and red photophores on its head. The dual-color bioluminescence is thought to serve both defensive and possibly hunting functions, though much about this species' ecology remains mysterious. The biochemical mechanism allowing the same organism to produce two distinct colors involves different forms of luciferase that modify a firefly luciferin in different ways. Beyond insects, terrestrial bioluminescence appears in fungi, creating another category of the glowing organisms on land. Bioluminescent fungi, often called foxfire, glow with a soft, greenish light that can be visible in dark forests. The glow comes from the mycelium, the thread-like fungal filaments, and sometimes from the fruiting bodies we recognize as mushrooms. Over 70 species of fungi are known to be bioluminescent, including members of the genera Armillaria, Mycena, and Pinellus. The function of fungal bioluminescence has puzzled scientists for centuries. Unlike fireflies, which clearly use light for communication, or marine organisms, which use it for hunting and defense, the purpose of glowing fungi is less obvious. Recent research suggests that the light may attract insects, particularly at night when the glow is most visible. These insects, landing on or near the fungi, may help disperse fungal spores, effectively functioning as pollinators for the fungi. The light production peaks at night and decreases during the day, supporting the hypothesis that it serves to attract nocturnal insects. The chemistry of fungal bioluminescence is distinct from that of fireflies or marine organisms. Fungal luciferin appears to be derived from caffeic acid, a compound common in plants and fungi. The luciferase is a multi-subunit enzyme complex, and the reaction requires a coenzyme to proceed. Despite these differences, the fundamental principle remains the same. An enzyme facilitates the oxidation of a substrate, producing light as a byproduct. A few other terrestrial organisms possess bioluminescence, though these are rare and often poorly understood. Certain springtails, tiny arthropods found in soil and leaf litter, produce faint glows. Some millipedes are bioluminescent, potentially using light as a warning signal like firefly larvae do. There are even reports of bioluminescent earthworms, including species in the genus Diplocardia, though the function of their light remains unknown. These organisms glow when disturbed, suggesting a defensive function, but much research remains to be done. Land snails with bioluminescence are extremely rare, but they do exist. Quantula striata, a snail native to Southeast Asia, is the only terrestrial snail known to be bioluminescent. It produces a greenish glow from its soft tissues, visible when the snail extends from its shell. The function of this bioluminescence is unclear, though it may serve in species recognition or as a warning signal. The relative rarity of terrestrial bioluminescence compared to marine bioluminescence likely reflects several factors. On land, there are alternative communication strategies that work well. Sound travels effectively through air and costs less energy than producing light. Visual signals using reflected light, like colorful plumage or patterns, are effective during the day. The abundance of ambient light during daylight hours reduces the effectiveness of bioluminescence except at night. In contrast, the deep ocean is perpetually dark, and sound, while it travels well through water, can attract unwanted attention. Visual signals using reflected light are useless where no light exists to reflect. Bioluminescence fills a critical niche in the deep sea that has no equivalent on land. The darkness of night on land is temporary, lasting only hours. The darkness of the deep ocean is permanent, creating stronger selective pressure for light-producing adaptations. Human impact on terrestrial bioluminescence, particularly fireflies, has become a conservation concern. Light pollution from cities and artificial lighting disrupts firefly communication. Males flashing their courtship patterns cannot be seen against the bright backdrop of streetlights and buildings. Females' subtle responses are invisible. As a result, firefly populations have declined dramatically in urbanized areas. Habitat destruction, pesticide use, and climate change add additional pressures. Some species of fireflies that once were common have become rare, and a few may face extinction. Despite these challenges, terrestrial bioluminescence remains one of nature's most accessible wonders. Unlike deep-sea bioluminescence, which requires specialized equipment and submersibles to observe, fireflies can be watched from your backyard on a summer evening. The soft, pulsing lights drifting through the darkness connect us directly to the chemistry and evolution we've been exploring, making the phenomenon tangible and immediate. Surface waters and dinoflagellates. Not all marine bioluminescence occurs in the deep ocean. Some of the most spectacular and accessible bioluminescent displays happen at the ocean's surface, in shallow coastal waters where anyone can witness them. These displays, often called bioluminescent bays or glowing beaches, are created primarily by single-celled organisms called dinoflagellates, and they transform nighttime waters into shimmering fields of light. Dinoflagellates are a diverse group of single-celled plankton, most of which are not bioluminescent. But certain species, particularly Noctiluca scintillans, and several species in the genus Pyrocystis produce brilliant blue-green light when disturbed. These organisms drift in surface waters, sometimes accumulating in enormous numbers during seasonal blooms. When conditions are right, trillions of these tiny organisms can occupy a single bay or stretch of coastline, creating what locals and tourists have described as seas of stars. The bioluminescence in Dinoflagellates serves a defensive function. When a wave breaks, when a boat passes through, when a fish swims nearby, or when a swimmer moves through the water, the physical disturbance triggers the dinoflagellates to flash. Each organism produces a brief pulse of light, less than a second in duration. But when millions or billions of dinoflagellates flash simultaneously in response to the same disturbance, the cumulative effect is spectacular, creating glowing waves, trails of light behind swimmers, or sparkles around the hulls of boats. The defensive benefit of this light production relates to the burglar alarm strategy we discussed earlier. When a small fish swims through a patch of dinoflagellates, attempting to feed on zooplankton or other tiny organisms, the disturbance causes the dinoflagellates to light up. This sudden burst of light may startle the fish, disrupting its feeding. More importantly, it makes the fish highly visible to larger predators. A fish illuminated by bioluminescent dinoflagellates stands out like a beacon in the dark water, attracting the attention of predatory fish, seabirds, or marine mammals. The fish faces a choice: continue feeding and risk being eaten, or flee the bioluminescent patch. Either way, the dinoflagellates benefit. The chemistry of dinoflagellate bioluminescence involves a luciferin, called dinoflagellate luciferin, which is structurally distinct from other luciferins. The enzyme is called dinoflagellate luciferase, and interestingly, it's related to chlorophyll-binding proteins, suggesting an evolutionary connection between photosynthesis and bioluminescence in these organisms. The light-producing reaction occurs in tiny organelles called scintillons, specialized structures found only in bioluminescent dinoflagellates. The trigger for light production is mechanical disturbance. When water flows past a dinoflagellate, when it's jostled by waves, or when it collides with something, mechanoreceptors in the cell membrane detect the stress. This triggers a cascade of cellular events that ultimately allows protons to flow into the scintillons, lowering their pH. The acidic environment activates the luciferase, which then catalyzes the oxidation of luciferin, producing light. The entire process, from disturbance to light production, takes less than 20 milliseconds, making it one of the fastest bioluminescent responses known. Dinoflagellate bioluminescence follows a circadian rhythm, a biological clock synchronized to the day-night cycle. During the day, the scintillons are more fully functional. The cells produce the necessary luciferin and luciferase primarily at dusk, preparing for the night ahead. Light production peaks during the dark hours and decreases again as dawn approaches. This rhythmic control ensures that the cells don't waste energy producing light when it wouldn't be visible or useful. The most famous locations for observing dinoflagellate bioluminescence include Mosquito Bay in Vieques, Puerto Rico, often cited as the brightest bioluminescent bay in the world, Luminous Lagoon in Jamaica, and various bays in Vietnam, Thailand, and the Maldives. These locations combine the right environmental conditions to support dense blooms of bioluminescent dinoflagellates. Warm water, shallow protected bays that concentrate the organisms. Limited tidal flushing that prevents them from being dispersed, and often input of nutrients from rivers or mangroves that fuel population growth. Visitors to these bioluminescent bays describe the experience as magical and surreal. Swimming at night, your movements create swirls of blue-green light. Every stroke leaves a glowing trail. Fish dart away, briefly illuminated as moving sparks. Splashing creates explosions of light. Rain on the surface can make the entire bay shimmer with millions of tiny flashes. The effect is otherworldly, like swimming through a liquid sky filled with stars. But these bioluminescent ecosystems are fragile. Pollution, particularly from sunscreen, bug spray, and other chemicals that tourists wash off when swimming, can harm or kill dinoflagellates. Light pollution from coastal development can disrupt their circadian rhythms and make the bioluminescence less visible. Coastal development that increases runoff or changes water flow patterns can alter the conditions that allow dinoflagellates to bloom. Climate change, with its effects on ocean temperature and pH, may shift the ranges where these organisms thrive. Several famous bioluminescent bays have experienced declines in their glow, and conservation efforts are underway to protect these unique ecosystems. Not all surface bioluminescence comes from dinoflagellates. Certain species of jellyfish produce light when disturbed. Small crustaceans called copepods can create flashes when they move. Even some species of fish that live in surface waters have bioluminescent capabilities, though these are less commonly observed by casual visitors. But dinoflagellates are the primary architects of the glowing waves and sparkling bays that have captured human imagination for centuries. Historical accounts suggest that sailors have long been familiar with bioluminescent seas. Christopher Columbus, during his first voyage to the Americas, recorded observations that may have been of bioluminescent waters. Ancient mariners spoke of phosphorescent seas, though they didn't understand the source of the glow. The term phosphorescent is actually a misnomer. Phosphorescence is a different physical process that doesn't involve living organisms. But the name stuck for centuries before scientists understood that the ocean's glow came from chemistry performed by living cells, not from minerals or other non-biological sources. The study of dinoflagellate bioluminescence has contributed significantly to our understanding of circadian rhythms, cellular organization, and defensive strategies in microscopic organisms. These tiny cells, barely visible individually, collectively create some of the most spectacular light displays in nature. They remind us that bioluminescence operates at every scale, from single-celled plankton to massive squid, from bacteria to fish, each organism using the same basic chemistry, but adapting it to its own specific needs and environments. Unusual and rare. Bioluminescent creatures. Beyond the well-known fireflies, anglerfishes, and dinoflagellates, the world harbors numerous lesser-known bioluminescent organisms, whose light-producing capabilities are no less remarkable for being obscure. These rare and unusual species expand our understanding of how broadly bioluminescence has been adopted across the tree of life, and how creatively evolution has deployed this capability. The cookie-cutter shark, which we've mentioned briefly, deserves fuller attention. These small sharks, typically less than 20 inches long, have one of the most unusual hunting strategies in the ocean. They possess photophores covering most of their ventral surface that produce a blue-green glow, allowing them to blend with downwelling light when viewed from below. Classic counter-illumination camouflage. But crucially, they have a dark, non-luminous collar around their throat that doesn't glow. When a larger predator looks upward at a cookie-cutter shark, the glowing body disappears against the ambient light, but the dark collar stands out. To the predator, this dark patch resembles a small fish, an easy meal. As the predator approaches to investigate, the cookie-cutter shark, which is perfectly capable of seeing its approaching attacker, positions itself carefully. At the last moment, the shark lunges and uses its specialized teeth to excavate a circular plug of flesh from the side of the much larger animal. The cookie-cutter shark's teeth are adapted specifically for this parasitic feeding strategy. The lower teeth form a saw-like arrangement that can cut through skin and blubber. The upper teeth are pointed and designed to anchor the shark to its victim. The shark bites, twists its body, and removes a chunk of flesh shaped like a cookie, hence the name. The victim, whether a dolphin, whale, tuna, or even the occasional submarine's rubber coating, is left with a distinctive crater-shaped wound, and the shark swims away with a nutritious meal. This hunting strategy represents an inversion of typical camouflage logic. Instead of trying to be completely invisible, the cookie-cutter shark uses camouflage with a deliberate flaw, making itself look like something else, something smaller and more vulnerable. It's aggressive mimicry taken to a creative extreme, using bioluminescent camouflage to enable parasitic predation. Bioluminescent earthworms provide another surprising example of terrestrial bioluminescence. Several species, including Diplocardia longa in North America and various species in South America and Asia, are capable of producing light. When disturbed or stressed, these earthworms exude a bioluminescent fluid from their pores or produce light within their tissues. The fluid can glow for several minutes, coating the worm or the surrounding soil in light. The function of this bioluminescence remains somewhat mysterious. It likely serves as a defensive warning, the worm's equivalent of a colorful pattern announcing unpalatability. Earthworms are prey for many animals, including birds, moles, shrews, and various insects. A glowing worm might startle these predators or advertise that the worm contains toxins or tastes unpleasant. Some researchers have suggested the light might also deter parasites or pathogens, though evidence for this is limited. The chemistry of earthworm bioluminescence is distinct from both fireflies and marine organisms. The exact nature of the luciferin and luciferase involved varies by species and remains an active area of research. Some earthworm species appear to have evolved bioluminescence independently of other terrestrial organisms, representing yet another independent origin of this capability. Scale worms, segmented marine worms found at various ocean depths, possess bioluminescent scales that can be shed when the worm is attacked. These scales continue to glow after detachment, creating a distraction while the worm escapes. Some species can produce particularly bright flashes, bright enough to temporarily blind or disorient predators. The scales eventually regenerate, making this a renewable defensive resource. Tomopteris, a genus of pelagic polychaete worms, produces what may be the only known example of yellow bioluminescence in the ocean. Most marine bioluminescence is blue or blue-green, as we've discussed, because these colors travel farthest through water. But Tomopteris worms, when disturbed, release glowing particles that emit yellow light. The function of this unusual color is unclear, though some researchers speculate it might be particularly effective at startling specific predators, or that the yellow light might appear brighter at close range, maximizing the startle effect. Among mollusks, most bioluminescent species are cephalopods, squid, and octopuses. But a few bivalves, the group that includes clams and mussels, are also capable of producing light. These species typically live in deep-sea environments and possess light-producing cells in their mantle tissues. The function appears to be defensive, with the bivalves producing flashes of light when disturbed by predators. Given their sedentary lifestyle, bivalves can't flee from predators, so startling them or attracting their predators' predators may be their best defensive option. The Hinea snail, a marine gastropod found in shallow waters, produces flashes of blue-green light through its translucent shell when disturbed. This is one of the few documented cases of bioluminescence in gastropods. The light appears to serve a defensive function, potentially warning predators that the snail is unpalatable or startling them at the moment of attack. Several species of brittle stars, echinoderms related to sea stars, are bioluminescent. When attacked, they produce waves of light that ripple along their arms, creating a spectacular display. If an arm is caught by a predator, the brittle star can detach it. The severed arm continues to glow and thrash, distracting the predator, while the rest of the brittle star escapes. The arms regenerate over time, making this a costly but effective last resort defense. Certain species of fish have evolved bioluminescent capabilities that seem almost experimental in their variety. The midshipman fish, named for the rows of photophores along its sides that resemble the buttons on a midshipman's uniform, can produce both light and sound. Males hum during breeding season, a low-frequency drone that attracts females while their photophores glow. The combination of acoustic and visual signaling is unusual among marine fish. The stoplight loose-jaw dragonfish deserves another mention for just how unusual its bioluminescent adaptations are. Beyond its red light-producing photophore and red-sensitive vision, this fish has a jaw that can unhinge and extend incredibly far, allowing it to swallow prey larger than its own head. The combination of invisible red light hunting with an oversized mouth makes it a supremely effective predator in its deep-sea environment. Barrel-eye fish, members of the family Opissoproctidae, have transparent heads that allow their upward-pointing eyes to see through their own skulls. Some species in this family have bioluminescent organs that may help them detect bioluminescent prey silhouetted against the faint light from above. Their entire anatomy seems designed around processing visual information in the dim light of the mesopelagic zone, and bioluminescence plays a role in this specialized sensory world. Among sharks, aside from the cookie-cutter shark, several other species are bioluminescent. The lantern sharks, named for their light-producing capabilities, possess photophores that may help with species recognition or counter-illumination. The pygmy shark, one of the smallest sharks in the world at less than 10 inches long, has a luminous belly that likely serves for camouflage in the twilight zone. The fact that bioluminescence appears across multiple shark families suggests it evolved independently several times within this ancient group of fish. These unusual and rare bioluminescent organisms remind us how much we still don't know about this phenomenon. Many species have been observed to glow, but the function of their bioluminescence remains uncertain. The chemistry of many bioluminescent systems hasn't been fully characterized. The evolutionary history, how and when these capabilities arose, remains speculative for many groups. Every deep-sea expedition discovers new bioluminescent species, expanding the known diversity of light-producing life. The creativity of evolution, if we can call it that, is on full display in these unusual bioluminescent organisms. Each has found its own solution to the challenges of its environment, its own way to make use of chemistry to produce light that serves some benefit. Whether it's the cookie-cutter shark's inverted camouflage, the earthworm's glowing defense, or the Tomopteris worm's unusual yellow light, each represents a unique evolutionary experiment, a biological innovation that works well enough to persist. The future of bioluminescence research. As we near the end of our gentle journey through the glowing world of bioluminescence, it's worth considering what the future might hold for this field of study. Bioluminescence research sits at the intersection of basic science, medical applications, environmental monitoring, and conservation biology. The insights gained from understanding how organisms produce light have implications far beyond the organisms themselves. In medicine and biotechnology, bioluminescent systems have become indispensable tools. Researchers use luciferase as a reporter gene to track gene expression in living cells and organisms. When they want to know whether a particular gene is active, they attach the luciferase gene to it. If the target gene turns on, the cells glow, providing a visual, easily measured signal of genetic activity. This technique has been used to study cancer, to track the spread of infections, to understand how embryos develop, and to investigate countless other biological processes. Bioluminescent imaging allows researchers to watch processes inside living animals without surgery or dissection. A mouse engineered to express luciferase in its cancer cells will glow wherever those cells are located. By measuring the light produced, researchers can track tumor growth, monitor how cancers respond to treatments, and study metastasis in real time. This capability has accelerated cancer research and led to the development of more effective treatments. ATP detection using Firefly luciferase has become a standard method for measuring bacterial contamination in food, water, and medical equipment. ATP is present in all living cells, so its detection indicates the presence of bacteria or other microorganisms. A sample is mixed with luciferin and luciferase. If ATP is present, light is produced with the intensity proportional to the amount of contamination. This test is fast, sensitive, and doesn't require growing bacterial cultures, making it ideal for quality control in various industries. researchers are exploring bioluminescence for optogenetics, a technique where light is used to control neural activity. While most optogenetic approaches use external light sources directed at genetically modified neurons, some scientists are developing systems where bioluminescence internally generated by cells could activate light-sensitive proteins, potentially allowing for wireless control of neural circuits. This could have implications for treating neurological disorders or for basic neuroscience research. The study of bioluminescent symbioses has shed light on how organisms interact with their microbiomes, the communities of bacteria that live in and on larger organisms. The Hawaiian bobtail squid and its bacterial partners have become a model system for understanding how hosts and bacteria communicate, how immune systems distinguish between beneficial and harmful microbes, and how these partnerships develop and maintain themselves. The insights gained apply to human health, where our own gut bacteria and other microbial partners play crucial roles. Environmental monitoring represents another application of bioluminescence research. Bioluminescent organisms are sensitive to pollution, changes in water chemistry, and other environmental stressors. Monitoring the health of bioluminescent populations in bioluminescent bays provides early warning of environmental degradation. Some researchers are developing biosensors using bioluminescent bacteria or other organisms to detect specific pollutants. The bacteria glow in the presence of the target pollutant, providing a biological assay that can be more sensitive and specific than chemical tests. Climate change impacts on bioluminescent organisms are an emerging area of concern and study. Ocean acidification, caused by increasing carbon dioxide levels in the atmosphere being absorbed by seawater, may affect the chemistry of bioluminescent reactions or the health of the organisms producing the light. Temperature changes can affect the distribution of bioluminescent species, potentially disrupting the ecosystems that depend on them. Coral reef systems, some of which include bioluminescent organisms, face multiple stresses from warming waters and acidification. Conservation of bioluminescent ecosystems has become a priority in some regions. The bioluminescent bays of Puerto Rico, for instance, are protected by law, with restrictions on development, boating, and swimming to preserve the fragile dinoflagellate populations. Similar protections exist for other notable bioluminescent sites around the world. However, many bioluminescent ecosystems, particularly in the deep ocean, remain vulnerable to human impacts from fishing, pollution, and climate change, often without legal protection or even scientific documentation of what exists. The deep ocean, where bioluminescence is most prevalent, is one of the least explored environments on Earth. We've mapped the surface of the Moon and Mars in greater detail than we've mapped our own ocean floor. Vast expanses of the deep sea have never been visited by humans or human-made vehicles. Every deep-sea expedition discovers new species, many of them bioluminescent. Who knows what remarkable organisms and what innovative uses of bioluminescence remain to be found? New technologies are enabling better observation and study of bioluminescence. Improved cameras sensitive to low light levels can capture bioluminescent displays in detail never before possible. Autonomous underwater vehicles can explore areas too deep or remote for human-occupied submersibles. Genetic sequencing allows researchers to identify the specific luciferin and luciferase genes in newly discovered bioluminescent species, helping to map the evolutionary history of these capabilities. Synthetic biology opens possibilities for engineering new bioluminescent systems. Researchers can mix and match luciferins and luciferases from different species, creating hybrid systems with novel properties. They can modify the enzymes to produce different colors of light or to respond to different triggers. Plants have been engineered to glow, though weakly, raising the possibility of trees that could serve as biological streetlights, though practical implementation faces significant challenges. Understanding the evolution of bioluminescence remains an active research area. With at least 40 independent origins of this capability, likely more, bioluminescence represents one of evolution's most repeated inventions. By studying the genetic and biochemical pathways that led to bioluminescence in different lineages, researchers can understand how complex traits evolve, how pre-existing genes are co-opted for new functions, and how similar solutions arise independently in response to similar environmental pressures. The cultural and economic importance of bioluminescence shouldn't be overlooked. Bioluminescent bays attract eco-tourists, providing income to local communities as well raising awareness of marine conservation. Firefly festivals in Japan and other countries celebrate these insects and their glowing displays, connecting people to nature and traditional culture. The aesthetic and emotional impact of bioluminescence, its capacity to inspire wonder and curiosity, may be its most important function from a human perspective, even if evolution cares nothing for human emotions. As we continue to study bioluminescence, we're reminded of how much remains mysterious. We don't fully understand the function of bioluminescence in many organisms. We don't know all the types of luciferin that exist, or all the ways luciferases can be structured. We're still discovering new bioluminescent species and new applications for bioluminescent chemistry. The glowing world we've explored tonight is more vast and more complex than we yet appreciate. And now, as our journey draws to a close, the story of bioluminescence comes full circle. From the chemical reactions occurring inside cells smaller than a period on a page to the vast light shows spanning kilometers of ocean, from the first organisms that stumbled upon this chemistry billions of years ago, to the humans using it to cure diseases and monitor environmental health. Bioluminescence connects scales and times and species in an intricate web of cause and effect. You've drifted through oceans of light and darkness, through forests where beetles flash codes to one another, through tide pools where snails glow warnings, through the chemistry that makes it all possible. You've met anglerfishes and vampire squids, fireflies and dinoflagellates, bacteria and fungi, each glowing for its own reasons, each carrying within it the same basic chemistry, adapted to 1,000 different purposes. The light produced by these organisms is not metaphorical. It's real photons, real electromagnetic radiation at visible wavelengths, real light that travels through water or air and strikes real eyes, triggering real neural responses. Yet it feels magical. It seems too beautiful, too purposeful, too eloquent to be simply the byproduct of evolution and chemistry. But that's exactly what it is, and that, que la merero, the more remarkable. As you rest now, as your breathing slows and your thoughts begin to drift toward sleep, carry with you this knowledge. The world glows. In oceans and forests, in caves and bays, in the deep places we rarely see, and the summer evenings we take for granted, life makes light. Not borrowed from the sun, not reflected from some external source, but generated from within. Chemistry transformed into photons, survival strategies written in bioluminescence. You are part of the same story. The atoms in your body were forged in stars, just like the luciferins and luciferases in a firefly. The oxygen you breathe is the same oxygen that combines with luciferin to produce light. You are made of the same fundamental materials, governed by the same physical laws, shaped by the same evolutionary processes. You, too, glow, though in infrared wavelengths your eyes cannot detect. Thermal radiation from the warmth of your living body. Tonight, somewhere in the ocean, bioluminescent organisms are flashing, glowing, pulsing with light. Fish are hunting with living lanterns. Squid are protecting themselves with clouds of glowing mucus. Bacteria in symbiotic partnerships are producing steady beams to help their hosts survive. And on land, in certain forests and meadows, fireflies are beginning their evening displays, males flashing their species-specific patterns, hoping to attract a mate before the night ends. The world is more luminous than it appears. Beneath the darkness, within the darkness, sometimes because of the darkness, life finds ways to shine. If these explorations of science and wonder have helped you unwind or given you a peaceful way to end your day, I hope you'll consider subscribing to the Sleepy Science Society and liking this video. It helps others find this quiet corner where science meets rest, one sleepy soul at a time. Sleep well, dream softly, and remember that even in darkness, even in the deepest parts of the ocean and the quietest hours of night, the world glows with a living light. Good night.